This is your Prasanna Ma'am from NEET World. Before getting into the concept of our 100 days topmost topics for NEET 2026, let me introduce in brief about NEET World special programs. For 11th standard students, you land on NEET World's 2 Steps to Medicine program beta, wherein you will be aiming for NEET 2027, and the fee is very minimal of 3333 per month, and then NEET World's 1 Step to Medicine, which is exclusively for 12th standard students, and the fees is again minimal, that is 3333 per month. And unique program for long term students, you land on the platform of NEET World's Powered Step to Medicine of the fee 3333 per month. So, any program it might be, it is the same fee. There is no doubt in the quality of lectures that you get and complete study material access, and undoubtedly you will be getting one-on-one mentorship for each and every student from the expert faculties of NEET World. These are the special programs from NEET World. For any kind of information from this, you need to call to this number given below and to the numbers which are scrolling here also. And if you want to visit the branch in offline, you can come here in the Narayanguda, Hyderabad. Clear? Next. And the new batch is going to start from July 1st, 2026. Okay. So, next if we have to look into biomolecules. In biomolecules, yesterday we have dealt with carbohydrates, where we have looked into monosaccharides, derived monosaccharides, oligosaccharides and polysaccharides. In that we have again looked into homopolysaccharides and heteropolysaccharides. Today we are going to learn about proteins. So, proteins, if I have to describe beta, proteins are nothing but they are the chains of amino acids. You can simply say that proteins are polymers of amino acids. Okay, proteins are nothing but what? Polymer of what? Amino acids. Fine. And undoubtedly, no protein is a homopolymer beta, because you have 20 different types of amino acids. No protein is a homopolymer. Proteins are what? Heteropolymers of amino acids. And here you look into the basic classification of amino acids. This is important not just with respect to biology, but for your chemistry also. Looking on to the types of amino acids, which are polar, which are not polar, and which are aromatic, aliphatic, positively charged, negatively charged. Okay? Look into that properly. Amino acids which are hydrophilic or polar. Hydrophilic means water loving or hating groups. Hydrophilic, they are water loving groups. Phobic, you remember like this beta. Phobic means phobia. Hydrophobic, they are water hating groups. So, this hydrophilic or polar amino acids, under that you can see negatively charged or acidic amino acids, which includes aspartic acid and glutamic acid. Easy to remember na? Then under hydrophilic polar amino acids, you will also learn about basic type of amino acids which are of positively charged lysine, arginine, histidine, and positively charged like serine, threonine, asparagine, glutamine and cysteine. Both are same only. Okay. So, both are what? Basic proteins, positively charged amino acids, hydrophilic polar amino acids. Okay. Of this lysine and arginine are the important compositions, or these are the main basic amino acids that you will see in which proteins? Histone proteins. Remember, these histone proteins are positively charged proteins. They are highly used in packaging of the negatively charged DNA, condensation of DNA, and this histone proteins are composed of what? Lysine and arginine. Clear beta? You are clear with hydrophilic or polar amino acids, right? Now, when you look into hydrophobic amino acids or non-polar amino acids, non-polar or hydrophobic, you can look into aliphatic side chain or aromatic side chain. Okay. Aliphatic side chain includes glycine, alanine, valine, leucine, isoleucine, proline and methionine. These are the aliphatic amino acids, aliphatic, hydrophobic, non-polar amino acids. Okay. Aromatic amino acids are just 3: phenylalanine, tryptophan and tyrosine. Fine ma? Easy, right? So, you can easily remember aromatic amino acids and you can easily remember acidic hydrophilic polar amino acids. It is easy to remember the histone proteins lysine and arginine, and rest all just you have a idea about that. Okay, on that you can choose the options easily. Here you look into aliphatic side chain, glycine, alanine, valine, leucine, isoleucine, proline and methionine. Simple. Fine. This flow chart though, it is very important beta. Okay. And look into the basic structure of amino acid. At the center carbon will be present. Okay, carbon. To this hydrogen bond is there, R group will be present, amino group and carboxyl group will be there. Carboxyl group, amino group, hydrogen, and at other end R group will be present. This 3 will be same for all the amino acid. The change is with respect to what? R group. That's it. Okay. Now, so here you see these are what which are given in our NCERT, where glycine is the simplest amino acid, where the R group is what? Hydrogen. Hydrogen. R group is what? Hydrogen here. And alanine, if you see, that is methyl group in alanine. R group is what? Methyl group. In serine, if you see, hydroxy methyl group is present as R group. Clear? Remember these 3 because we can expect a direct question from here. Next, this should be clear in your mind about essential and non-essential amino acids, and these are the list of essential and non-essential amino acids. You remember one list and the other list you can easily identify. Okay, you remember the essential amino acids like phenylalanine, valine, tryptophan, threonine, isoleucine, methionine, histidine, arginine, leucine and lysine. Then it is easy to identify non-essential. Just one column you remember. Okay. Now, Zwitter ionic forms which are only applicable to the amino acids of protein. What is a Zwitter ion?
Contains positive and then negative.
Yes, which has both positively charged and negatively charged groups. Just look into the definition once. A Zwitter ion is a molecule with functional groups of which at least one has positive and one has negative electric charge. So, here if you see carboxylene is having negative charge and this is having positive charge. Zwitter ion is this, understood, right? If you are lowering pH than this, then this turns out to be what? Cation. Sorry, cation. On still lowering the pH, this turns out to be the Zwitter ion. And here if you increase the pH further, that turns out to be the basic form anion. Anion. Okay, look into that properly. Anion and cation. Zwitter ion with respect to cation and anion. Okay, so this is about Zwitter ion. In our questionnaire sessions, we will solve good questions about Zwitter ion. Next, here you see the peptide bond. We have discussed proteins are nothing but what? Chains of amino acids. 2 amino acids are connected by what? Peptide bond. Peptide bond is formed by carboxyl end of one amino acid and amino group of the other amino acid. By eliminating again what? Water moiety. Yes, dehydration we call it as. Okay. So, peptide bond is formed between carboxyl group of one amino acid and amino group of the other amino acid, and this is by elimination of what? Water moiety. The first amino acid is also called as terminal amino acid. In the complete chain of the amino acid, first amino acid is called as what? Terminal amino acids, N-terminal amino acid, and the last amino acid is called as what? C-terminal amino acid. Remember that. In a polypeptide chain, if I have to consider this as a polypeptide chain, okay, this first amino acid is called as what, Nana? N-terminal. Okay, N-terminal amino acid. Last is what? C-terminal amino acid. Okay, carboxyl terminal amino acid. As such, you can identify protein at 4 levels. It is having primary structure, secondary, tertiary and quaternary structures. This is very important. Okay, look into that carefully. First, primary structure, if you see, this is like a sequence of proteins, ma'am. Yes, only one sequence of peptide chain will be present, as how we have seen, like the first is having N-terminal end, last will be having C-terminal end. As such, the sequence of amino acids are just giving the positional information about the amino acids, and which is the first amino acid, which is the second amino acid, that positional information will be clear with this what? Primary structure of the amino acid. So, it is just imagined like a line, okay. It is imagined like a line, and the first amino acid and the right end, first amino acid is represented by the right end. Right means you can see from this view, from right end the first amino acid is represented, and last is what? Second, last amino acid, this is represented by what? C terminal, okay. First amino acid is also called as N terminal and last amino acid is called as C terminal. So, how you will remember about the primary structure of proteins? That is just a line like structure, okay, and that will give you the positional information about the amino acids, like which amino acid is first and which amino acid is last. Towards which end N terminal is present? Right end. Towards which end C terminal is present? That is it, okay, clear, right. Now, here you see secondary structure of protein. Secondary structure of protein is represented by two things, beta, alpha helix and beta plated sheets. Yes, alpha helix and beta plated sheets. So, here in the next slide clear information is given, but as of now you look into that clearly key. Alpha helix is nothing but the right handed helix that is formed because of the intramolecular hydrogen bonding, okay. This is alpha helix, which is just giving the helical structure, that is right handed helix, whereas beta plated sheets are formed as such key from one atom of one molecule with intramolecular hydrogen bonding, and the other atom of the molecule is highly electronegative atom, okay. Between those two atoms of different molecules bonds are formed, and that is forming what? Beta plated sheets. This two will come under what? Secondary structure of proteins, which gives you two dimensional structure of proteins. So, here you see the right handed helices which are observed, those are called as what, ma? It is symbol, those are called as what? Alpha helix. And in alpha helix you can see carbonyl of one amino acid is hydrogen bonded to amino hydrogen of an amino acid, that is 4 down the chain, nothing but every 4th amino acid is being bounded like this, okay. Between what? Carbonyl group and the amino group. Can you see clearly, ma? Carbonyl group and amino group, that is being attached like this. And next, this pattern of bonding will pull the polypeptide chain into a helical like structure, right. Every 4th amino acid, he said, you look into this properly, beta. Here 1, 2, 3 and 4, 4th amino acid. First and 4th are binded, no? By what? Carbonyl group and amino group. What is this bonding? Intramolecular hydrogen bonding. This is what bond? Hydrogen bond. Same the case, first and 4th amino acid will be binded. As such, those amino acids will be pulled and that will be forming what chain? Helical chain. Clear with? And this gives you what? Curled ribbon like structure. You have clearly understood now alpha helix. Now you look into the beta plated sheets. I have extracted the direct lines from NCERT, so that you should not get confused when you read the NCERT, okay. Beta plated sheets, 2 or more segments of the polypeptide chain, okay, 2 or more segments of the polypeptide chain will line up next to each other. They will line up next to each other like structures held by hydrogen bondings. Here also hydrogen bondings, but what I told you, hydrogen bonding of one atom of the molecule is binded to what? Highly electronegative atom of the preceding molecule, okay. This is all about alpha helix and beta plated sheets, okay. About beta plated sheets still, in addition to the long protein chain is also folded upon itself like a hollow woolen ball, giving rise to tertiary structure, okay. So, alpha helix, beta plated sheet are of secondary structure. If it is further folded like hollow wool like structure, that gives you what? Tertiary structure. So, this gives you what dimensional view? 3-dimensional view of protein. Tertiary structure is absolutely necessary for many biological activities of proteins, and the tertiary structure is mostly seen in the proteins like myoglobin like that, okay. So, here R group interactions will include hydrogen bonding, ionic bonding, dipole-dipole interactions. There only hydrogen bonding in the secondary structures, but here how many interactions are involved? Hydrogen bonding, ionic bonding, dipole-dipole interactions in the tertiary structure. Can you look into here beta? Amino acids which are of the primary structure, helical, alpha helix and beta plated sheet secondary structure, and into this hollow wool tertiary structure. Best example I can give you for alpha helix is keratin, keratin protein of hair, that is of alpha helix only, and collagen I can give you the best example for beta plated sheets. Remember that, okay? Now, so you look into here quaternary structure, okay? Tertiary structure is clear to you, right? Now, quaternary structure is observed in those proteins which have subunits. Tell me any proteins which have subunits? Very good. Hemoglobin, you can also say like myoglobin also, right? Hemoglobin, myoglobin which has different protein subunits, those are included under the quaternary structure. So, some proteins are an assembly of more than one polypeptide subunit. Hemoglobin is the best example of quaternary structure, wherein adult hemoglobin will be having 4 subunits. So, here you can see just 2 subunits, wherein inside that you can see alpha helix and beta pleated sheets. But adult hemoglobin which has what? 4 subunits, completely tertiary wool structure you can see, right? And somewhere you can see beta plated sheets and alpha helix as well. In quaternary structure you can see primary, secondary, tertiary structures, all the structures you can see. Okay, so this is the tertiary quaternary structure with 4 subunits. Clear right now beta? This is all about primary structure, secondary structures, tertiary and quaternary structure of the proteins, types of different structures of the proteins. Now at the end, let us look into the different types of proteins and their function, highly important. Okay, collagen, most of its structure is of what type? Beta plated sheets. Okay, it is mainly used in what? Intercellular ground substance, matrix region it will form. Next, trypsin will act as what? Enzyme. Okay, it will mainly lyse what? Biomolecules? Proteins. Yes, proteins not into peptides. Yes, okay, it will simplify proteins to what? Amino acids, trypsin. Next, insulin which acts like hormone, this is also a protein. Antibody type of protein, what does it do ma? Functions against antigens or any foreign particles. Pathogens you can call it as, foreign particles you can call it as, infectious agents also. Next, receptor for sensory reception like smell, taste, hormone. Okay. Next, GLUT4. GLUT4 is nothing but the transport, glucose transporter pump also you can say, glucose transport it will function as. This enables glucose transport into the cells. Clear right Nama? Now you look into the major functions of the proteins. Some proteins will transport nutrients across the membrane. Across the cell membrane you can see carrier proteins, channel proteins which facilitate what? Facilitated diffusion. Okay. So, that is one of the primary function of the proteins, that is what? Transport. Okay. And some hormones and enzymes will fight against the infectious agents like antibodies. Collagen is the most abundant protein in the animal world. Collagen is of what structure? Beta sheath. Rubisco, that is ribulose bisphosphate carboxylase oxygenase. This is the most abundant protein in the whole biosphere. Fine. This is synthesized from plants. Okay. So, with this we can end the proteins. One thing I would like to say you about Rubisco connecting to the plant physiology. Rubisco is synthesized where? Where is Rubisco synthesized? Think. Yes, inside the chloroplast Rubisco is synthesized. If I can consider this as a chloroplast, inside the chloroplast Rubisco is synthesized in stroma, wherein we call chloroplast as self-replicating organelle or semi-autonomous organelle, because chloroplast is having its own DNA. That DNA is of what type? Double-stranded circular DNA.
Primitive DNA.
Yes, double-stranded circular DNA. What is the ultimate goal of DNA? Any DNA it might be. What is the ultimate goal of?
Information.
Ultimate goal of DNA is protein synthesis. Okay, as such, the double-stranded circular DNA which is present in the stroma of the chloroplast, that will be synthesizing this world's abundant protein rubisco. Okay, this rubisco is being synthesized in the stroma of the chloroplast by what DNA? Double-stranded circular DNA. Okay, in this semi-autonomous organelle. Little connecting dot which I would like to say. Okay. So, with this we can end today's session, but once again I would like to brief you all the students about NEET World's special programs of 2 steps, 1 step and power step 2 medicine. For all the students of different financial backgrounds, this is the best place where this is the most minimal fee in the entire market of 333 per month with complete materials and one-on-one mentorship in all your journey, be it NEET 2026 or the target is for NEET 27, NEET World will be for your complete journey, okay, and the fee is this much only, and you can call us on to the given number for more information about these special programs, or you can directly visit the branch at this address. The batch will start at July 1st. Okay, and one more thing I have to say you is Telegram group. You know about this Telegram group? Join the Telegram group today only. What is the benefit about this Telegram group is, you will be having daily quizzes, okay, for botany, zoology, physics, chemistry and mathematics also, whoever are aspiring for JEE Mains. Okay, daily quizzes will be there in this Telegram channel of NEET World. Second is live class schedules will be updated in this Telegram channel. Third is free resources, online study materials will be provided, online questionnaire materials will be provided. Third is one-on-one doubt solving faculty will be provided. In call you can ask us any doubts, or you can drop a message in the Telegram channel also, we will be responding. For more information about the Telegram channel, you can call us to the given number. You can get into the Telegram group by logging on to this website or directly scanning this QR code. Okay. So, here the NEET World will be your ultimate destination for NEET, JEE, EAP set and CUET entrances. So, do not miss this opportunity, be with us, in touch in the Telegram group and explore your subject each and every day. Thank you, meet you tomorrow.